Hello, everybody, and welcome back to Creation Myths. We are here tonight for what I think will be a really fun and, if history is any indicator, probably just like a really nerdy show. I'm really excited. I've got Sal Cordova here with me, and we are going to talk about protein orchards, which is to say the separate ancestry of different protein families and the implications of that for evolutionary theory. Before we get going, let me just take a second here and tell you that Sal is an old hand in the evolution, creationism, ID stuff. He's been doing this for decades. You're probably familiar with his name. And okay, good. YouTube sounds good. And he has more degrees than I can ever remember. So Sal, if you want to, share your credentials, feel free. But I can never remember each specific degree that you have. So I'm just going to turn it over to you. Take a minute, introduce yourself, and then turn it back to me for just a second, and then we can pull up your slides, and you can, take a few minutes to make your case when it comes to protein orchards. But first, tell us about you.
I'm a molecular biophysics researcher, and I'm also a PhD student in biomolecular engineering. My prior life was a senior engineer and scientist in the aerospace and defense industry. And then I worked for Dr. John Sanford while I was taking care of my widowed and disabled mom. And so I used to be an evolutionist. I was raised in a Roman Catholic home. And then I became an evangelical Christian. And then somewhere along the way, it took about 30 years to go from evolutionist to old earth creationist to ID proponent to young life creationist, then young earth creationist. So I'm really honored to have a chance to make this presentation and this discussion. And I asked Dr. Dan if I could gush over him a little bit. Could you show the slides? I have a list of my favorite evolutionists. I didn't know, two of them are at Rutgers University, like Dr. Dan. I didn't know that Cy Gart is a visiting professor, and then also Helen Fisher. So these are my favorite evolutionists. And Dr. Dan, this is like 4 years ago, we had a debate, and there's also Erica there. And one reason I'm here tonight is Dr. Dan has actually given me valuable editorial feedback. I've corrected a lot of things in the way that I teach ID and creationism because of him, and just some examples. He taught me a little bit about phylogeny. Oh, by the way, Dr. Dan, when there was a poll of graduate, creationist science students, biology students, they said, who would they rather have, Dr. Dan or Kent Hovind? It was Dr. Dan unanimously. And here is Dr. Dan correcting me on something with Jeff Tompkins. Oh, you wanted to say
I just want to say, I should hope so. I hope that's the case. I hope so.
So like Dr. Dan will watch my channel and say, Sal, that's not right. And we'd talk about it. And they say, well, Dr. Dan, you're right. I have to retract that. So this is one example we had to show on something Jeff Tompkins gave me from ICR, and Dr. Dan kind of corrected me. I wanted to express my gratitude. Recently, he came on my channel like 2 weeks ago. He said, Sal, you're so much better than phylogeny of cars. And we'll talk about that because that relates to protein orchards. I adjusted this talk today just because of that comment. I said, Dr. Dan's right. So I came up with it, and we'll talk about Owen's homology. In our debate 4 years ago, this graph was prominent. I said, Dr. Dan, I'm going to have to work on that. And so, and there were it led to genetic entropy 2.0. And Dr. Dan had confronted both Michael Behe and Otangelo Grasso. And I said, I think Dr. Dan's right. Irreducible complexity needs more qualification to be effective. So, by the way, Erica is also one of my favorite evolutionists. We may or may not have, if we have time, I actually presented on her channel. It wasn't a debate. It was just a friendly discussion about molecular phylogenetics, and that may have some relevance to what we talk about today. You could take that down. So I just wanted to say I'm very appreciative. This has been a very good relationship, and we'll see where we can agree. And, if we disagree, I would very much, I feel it's a matter of conscience that other people get to hear the other side. And I feel Dr. Dan represents the other side well. So I just had to say that, Dr. Dan, just my expression of appreciation. We've had a 4-year relationship, and I've been very grateful for it.
I will say, first of all, Thank you. I really appreciate hearing that. And I will say, Sal, we've had way longer than a 4-year relationship. We've had a 4-year relationship chatting on YouTube, but for people who don't know, we go way back to Reddit. And I mean, I was reading Sal's work and occasionally commenting way back in like the Panda's Thumb and Uncommon Descent blog wars days. So we go way back. So Sal, the topic tonight is this idea of protein orchards, and I've been wanting to talk to you about this concept because if I recall, you gave a talk at a recent creation conference on this topic, and basically you're the guy for this argument. Like, if you want to talk about irreducible complexity, you talk to Michael Behe, right? If you want to talk protein orchards, you're the guy making this argument. So I'm really happy to have you on to be able to kind of work through this. And I will say this, you've been making your rounds talking about this. So Real Science Radio has, you've been doing some interviews with them. I've watched the recent ones. The most recent one that they posted was on this exact topic. So I want to hear you make this case. Take 5 minutes, 10 minutes, make this case about protein orchards and evolution, and then we'll talk about it, everybody. That's the plan for tonight.
Okay, great. I actually think we might get some agreement. And Dr. Dan, I made a presentation at Liberty University Creation Research Society, and I actually featured a video of Dr. Dan. I spoke of you, reverently, just so you know.
Thank you.
I usually don't speak reverently of most evolutionary biologists, but I do of you, just you and Joe Felsenstein. So if we could bring the slides up again, this relates to how some of this developed was I was working on various publications. I recently, and Dr. Dan gave me permission, just a little bit of self-promotion. I published a peer-reviewed research article as a co-author in Oxford University Press. And some of the stuff that is in that article was kind of in the area of research, structural bioinformatics, that kind of motivated what my thesis tonight. One of the co-authors is Joe DeWeese. He's published in the journal Nature. He and I also published in Creation Research Quarterly. We've published like in FASEB. And then also, some of what we do tonight will actually relate to a Springer Nature reference chapter on fitness maximization. This is probably where Dr. Dan and I will disagree, but let's go to where we might agree, just to clarify some things of what I'm saying and what I'm not saying, because it's been mischaracterized on the internet, and it's hard to get people to untangle things, and I understand the reasons why. I just want to point out there are people in the ID community that are ambivalent, or they believe in common descent. Michael Behe said he accepts common descent. Richard Sternberg, he's still in evolutionary biology. He probably accepts common descent. Michael Denton. So these are names you might be familiar with. I'm actually, for the sake of this talk, going to say, let's assume common descent, organismal common descent. But that's different than the descent of all proteins. Now, this was just from a debate. This is really funny. Aaron Raw wrote this to me, and he consulted you. And I said, look, this is something I think we're actually all going to agree on, that proteins/genes do not have a common ancestor. So again, I want to say this is not a criticism of universal common ancestry. Absolutely not. Now, people know I'm a young Earth creationist, but I think this is the wrong argument to use for young Earth creationism. And I actually think some of the evidential case for young Earth creationism isn't where it needs to be. And so I just want to be on record. That's why you don't hear me talk about it that much. I think Dan's side of the argument, I would have a hard time defeating it. So I'm just going to assume universal common ancestry. Also, I've been accused of saying that's what evolutionists believe, that all proteins descended from a common ancestor. I'm like, no, I never said that of anyone currently. So I'm not accusing anyone of saying that. But it's kind of aggravating that I get mischaracterized. I'm trying to just set the record straight. There are not many Google hits or evolutionary papers that I find that talk about the protein orchard. So evolutionary biology is more than the assumption of common descent and inferring phylogenies. Here's some example titles where you could actually explore the questions that are active. So I'm not criticizing common descent, and I'm not criticizing phylogenetic methods as being valid or invalid. I'm talking about the mechanisms of evolving proteins. And these are some papers that Dr. Dan may be familiar with. There is the word homology, and currently homology, and Dr. Dan probably understands this better than I do, it's very tied to common descent. That wasn't the original version of homology. Richard Owen, who coined the word, said the archetype represented an idea in the divine mind, which also foreknew all its modifications. So I'm gonna use the word, just kind of for the sake of this discussion, ONS homology, to distinguish it from the evolutionary common descent notion of homology. It's just similarity. So we could say, and again, this is a criticism Dr. Dan fielded. He said, don't use that phylogeny. I said, okay, I'll call it ONS homology. And since it's just ironic, Dr. Dan and I were talking about cars backstage. I didn't know he likes Formula One and IndyCar racing. And we even talked about drag racers. Oh my goodness. We almost didn't start the show because we were talking about cars. Anyway, all cars share kind of a basic homology in terms of similarity. Obviously, it's not by common descent, but they have a similar archetype or architecture. This does, and then you can see there are other objects that share homology in the kind of at a gross scale. Like helicopters share homology with other helicopters, same for blenders, same for pianos, architectural. However, that does not, even batteries will share homology with other batteries, 12-volt batteries. But there's that ONS homology does not extend to the parts of the car. And let me see here. So I'm going to apply this thing about the parts. So let's assume all life has the same basic homology, meaning Virchow's principle. They're all cellular based if we define life as cellular life. So let's look at now proteins. I'm going to make kind of the similar argument. So this is the spelling of collagen and amino acids, about 1,500 amino acids. We are about 20 to 30% by weight proteins. And there is a pattern here, very striking. Every glycine amino acid is highlighted. It's a striking pattern. Then we could take the human zinc finger 136. We also see a striking pattern here. And we can have higher order where we group them in terms of the amino acids, the classes. You could see these higher order patterns here. And it's very easy to see just in terms of the spelling, what we call the primary structure of proteins, there's no homology. If you want to say, well, okay, there could be, be a common ancestor, but you can't demonstrate it. And so at the spelling level, that's true. Also at the, in terms of the shapes of the protein, so you could see here, this is the collagen shape. And the zinc finger doesn't quite have a really good representation of shape. So the spelling is different, the shape is different. So this again is like, there's no homology, and we would assume then that there is no common ancestor, at least you cannot demonstrate it because of the lack of either ONS homology or just plain vanilla homology. Any of the bioinformatic tools will also demonstrate this in terms of the sequence spelling. So you could take that down for now, and then I'll just kind of, maybe we could just pause here. And I say, I began to see this as I was going through bioinformatic tools and studying shapes. And I said, there doesn't appear to be a common ancestor for all major protein families. So Dr. Dan, if you wanted to comment.
Yeah, so on that we agree. As you said, like we agree that proteins, unlike cellular life, do not share universal common ancestry, right? We disagree about cellular life, but we agree that proteins don't share universal common ancestry. And so kind of I have laid out here, once you're kind of all done making your case, I'm going to go through and make sure that I can accurately kind of represent your case. But the first part is that genes do not all share common ancestors. You can have gene families where within the gene families you have common ancestry, but you have independent gene families that through the process of evolution, right, if you have universal common ancestry for cellular life, over evolutionary time, many, many, many gene families would have originated independent of each other. Is that?
Yeah. So the way I would characterize that, it's like, well, how could that be if you have a universal cell? And I just say, okay, in genetics, you have gene loci or a locus. And I would just say the way I would try to characterize your position would be, and then also in my old Earth model, even though I'm a young Earth creationist, if I had an old Earth model, by the way, Stephen Meyer may actually, he's a progressive creationist, so he's very, at least the last time I interviewed him, so it's very similar to Behe. You would have independent locations in the gene, and that's how it would be. That's how it would resolve how we could do that in a model where you have a organismal common ancestor, but then independent protein families. So we're in total agreement there. So maybe we could quit because now we're actually in agreement.
Good. Quit while you're ahead.
So
I think it's the next step is where I'm going to disagree. So, if you want, I can pull the slides back up, and then I think it's the next bit. That's where I'm going to.
My thesis then.
So here you go. Keep going.
All right. So I say that the creationists are very quick to invoke supernatural miracles or metaphysical miracles. I'm just going to say, okay, let's set aside these philosophical things. We could at least say in principle, from a statistical standpoint, what events we would consider like very, I wouldn't call it a miracle in the medical physical sense necessarily. I'll just say astronomically far from normal expectation. So if you have 500 coin, if you have a flip a coin that's fair and it's heads 500 times in a row, that would be astronomically far from normal expectation. Whether it's a miracle of God or not, that's different than the actual statistics of it. Separate question. And this relates, we are dealing with the question of evolutionary fitness and really in the last part, because most of this was about Fisher's fundamental theorem of natural selection. But at the very last part, we are questioning fitness, and that relates to evolvability, how well, effectively, Darwinian so-called natural selection can evolve something. And it was after we published that, I started to find all these other papers like selection-driven gene loss in bacteria, mutated genomes decay despite sustained fitness gains. I was seeing a decoupling between, well, I would expect that natural selection should be building genes. And I mean, that was the way I originally understood the theory. But as I studied population genetics, I said we ought to see a lot of loss. And I began to see experiments to that effect. You've done a video about me redefining fitness. And I just wanted to say, I'm not the only one that has, it's not just the creationists that have made the objections. I probably went farther than the evolutionary biologist. Lewontin's very respected. This is a great article, The Confusions of Fitness. I go farther than he does, though. And Andreas Wagner, also a very respected evolutionary biologist, he said, fitness is difficult to define properly. Taken together, these difficulties mean an unassailable measurement of an organism's fitness. Organism's fitness does not in practice exist. And I'll go into that a little bit more. Can
I just say on that, like, I disagree. And like evolutionary biologists like disagree with each other. I disagree. I think it's pretty easy to I mean, it's not necessarily easy in a technical sense to measure fitness, but like in principle, it's not a hard thing to do. Fitness is one individual's genetic contribution to the next generation relative to the other individuals in that population. That's your fitness. And like, you can measure this a bunch of different ways depending on the system. Like for me, looking at viruses, like all my grad school work was experimental evolution of bacteriophages, viruses that infect bacteria. And when you're dealing with viruses in a petri dish, you measure doubling time. So I can look at different populations and measure doubling time, and I could actually compete them against each other and see which ones would win. And like, it was really easy to measure fitness in that context. You figure out how to do it. You do the you do the math, and like you can do it. And depending on the organisms you're looking at, measuring one subgroup's or one individual's relative genetic contribution to the future compared to other individuals or other subgroups can be a hard thing to do. Like, just in a technical sense, that's a hard thing to do in many cases. But like, in principle, that's what fitness is. And I'm sure, there are obviously biologists that disagree with that definition of fitness, but like, you take what, as defined by Darwin, and you update that definition for, modern molecular genetics. That's what fitness is. It's you're using, looking at allele frequencies and such, and what's your contribution relative to everybody else, right? That's what that is.
So thank you for pointing that out. And I actually, in this is a paper from a smallish, ID-friendly journal. You could see that I actually go through kind of like what Dr. Dan was describing, but also kind of the formalisms of how it's traditionally done. Now, I just wanted to point out something. Andreas Wagner, again, in his book Robustness, he said, however, fitness is hard to define rigorously and even more difficult to measure. An examination of fitness and its robustness alone would not yield much insight into the opening questions. Instead, it is necessary to analyze on all levels of organization the systems that constitute an organism and that sustain its life. I define such systems loosely as assemblies of parts that carry out well-defined biological functions. And this is very similar to Michael Behe, a single system composed of several well-matched interacting parts that contribute to the basic functioning of the system. And I'm gonna show why I think that the standard, let's call it the standard definition of evolutionary fitness, does not actually solve the problem that Darwin is trying to solve, which is the emergence of organs of extreme perfection complication, Origin of Species, chapter 6. So again, we have, like you said, the doubling time. That's an exponential function, and you can relate that to absolute fitness. You can scale it. This is in my Blythe paper. It's an educational paper. It wasn't meant to be a research article. The problem that Lewontin articulated in the Santa Fe Winter Bulletin, and one evolutionary biologist said when he read that, he rejected Darwinian evolution from that point on. And I'm just extrapolating a little bit because Lewontin had this density-dependent fitness. He said, well, then what is fitness? Because if the environment changes, so like, actually, Dr. Dan, just really, if you could take this down for just a moment. I just have a technical question. It's actually really hard to tell what alleles affect a trait. I was looking at the peppered moth, and like the debate on which gene locus and what actually. It took a lot of physical. It's not easy research to identify the location and what gene actually is controlling what. So in one environment, I guess the light-colored moths would be favored. They would have this selective advantage. And then in another environment, it'd be the dark-colored. And this is one of the problems that Lewontin, in density-dependent selection, he said, well, one thing that changes the environment is just the number of organisms that have an allele. And it kind of bothered him. When we tried to resolve the equations, he said, this is just hard. If you could bring up my slide again.
Yeah. You have like frequency-dependent selection and density-dependent selection are things that exist, yes. And that changes the fitness landscape around. Yeah, these are things. But Sal, how does this relate to protein families and
why they're statistically improbable?
I'm
sorry
to have
to
get what I thought we were getting to.
I do apologize. I'm going to get there. I'm going to point out, usually the thing is that what they'll say is selection will solve the problem. This problem of the uncertainty of what's going to be selected for in any given environment, and you don't know what environment, you can't affix. If you can't even affix a probability of that, how can one assert that selection is going to evolve something? So one thing I know, this is from Bruce Albert's Baby Cell, Essential Cell Biology. This talks about how proteins can complement each other both in shape and charge distribution. So the way you spell one protein has to be able to be, if you have another protein that it is complementary to it in the course of a, particular eukaryotes, you have many large complexes. They have to complement each other in terms of their shape and also charge distribution, so they could find each other and connect in the context of a cell. So the connectivity is, again, similar to this nut fitting the bolt, except in protein, sometimes you're talking about angstroms, which is less than the radius, an atomic radius. So this is just a kid's toy, and and we could also see how all the pieces fit here precisely. So this relates again to kind of the engineering. I don't know how else to call it, maybe the engineering notions or geometric notions of what is fit. And this goes back to, let me see, I don't think I have that picture where we're talking about the Kimura distribution. See, this is independent of the environment, whether, they would make a good fit under some physiological conditions. And so this is why when you have a random mutation, it can really damage something very easily, because it's going to disrupt the way these parts fit. I felt that was a better notion of what we see when we talk about, I try to discourage the use of the word deleterious in this context, but that's just kind of just like, well, that's what would be a really bad mutation. It's so easy. But Michael Lynch said there's many more ways to break than there is to make. And that kind of illustrates the problem. So things, to evolve a major protein family, like the topoisomerase, and let me skip some more things here. It has to fit really well. There's 65, you could take this one down for the time being, this slide. Just for that topoisomerase to connect to, it's a homodimer. And when the parts connect, I did a X-ray crystallography study in silico. I didn't actually do the X-ray crystallography. Someone else did the hard work. When I ran it through a bioinformatic tool, there were like 60 connection points. I'm just like, it had to be geometrically positioned. And then some of these had to be, again, at close to the angstrom level of closeness. This is how tightly, it's so easy to disrupt that. So kind of just as a qualitative argument, how can natural selection build all these, homomeric, these heteromeric, or these protein complexes, when they just wouldn't form? And so the standard answer is natural selection will build it. And I'm saying at the very least, you can't make that assertion. That's kind of a faith statement, given we actually don't know what environments are going to select. And there's some experimental evidence that's kind of negative to the way we've conceived of selection.
Okay, thank you. So I'm going to try to steelman this argument. I'm going to try to make a short, and you let me know if my representation of this argument is accurate to the case you're making. You let me know. So we agreed that we're assuming, for the sake of this conversation, we're assuming that all cellular organisms share common ancestry. And then within that, there are gene families that do not share common ancestry. So these genes, those genes, they at some point in evolutionary history have independent origins through evolutionary processes. And within the gene family, you have common ancestry, but across the different gene families, they are unrelated to each other in terms of their evolutionary origins. Now, going from there, we have, in order for that to happen, you need to then have new genes, and new gene families have to evolve. But de novo gene origin is a sufficiently improbable event, a statistical miracle is the term you used, and the number of such events that would have to happen over the course of evolutionary time, from the first population of cells to all of the things that exist today, that number of de novo gene origins is so high that it is the idea right? So the argument says the probability of a single origin is so low and the number of origins required is so high. You put those two things together, that universal common descent of cellular organisms is not a feasible model, and separate creation, where the gene families are created intact, is a better explanation. Is
Actually,
that a fair presentation?
you were pretty close, and part of the reason it may differ from what I wanted is that's on me. Okay, so this is why it's good for us to discuss, because sometimes I definitely do not express myself well. Sometimes I fumble with my words.
No, I think you're good. This is easier than like going back and forth like via YouTube videos or like blog posts or something. This is easier. So please, clarify. Let me go through that little bit by bit, and you tell me what I should change. So we agreed on cellular common ancestry, but not protein common ancestry.
Right.
And then the origin of new genes or gene families is extremely improbable.
Okay, so let me stop there. I need to clarify. I wouldn't say all, and that's on me. That's on me. I should have, actually, I got to say, this is why I like interacting with you, because you kind of put my feet to the fire and say, Sal, you didn't really word that well. And even in the last week, I realized I need to amend that, especially, and I give you credit for alerting me to that. And I should have said it. It just slipped my mind. I would say there are some. It is. Okay, is Otangelo learned the hard way? You don't say all. Okay.
Okay. So I've amended what I wrote down then to say some instances of de novo gene origin are sufficiently improbable, parentheses, statistical miracle.
Okay, and I got to credit you, Dr. Dan, because the way I think you just kind of totally dismantled Otangelo. I said,
thank you.
Do not say all, because Dr. Dan's going to just make mincemeat of you. So and I have interest, it's like, I have interest in giving my side good arguments, and I think you'd be slaughtered. I mean, guys on my side, you'd be slaughtered if you said all. So I'm going to say there's some, and the some are the ones I'm familiar with. I cannot possibly explore all protein space and say, well, that's too hard to evolve. That would be too hard. Tangelo found out the hard way what happens when you make a sweeping statement. Someone's going to find an exception, and you did. So, and then also you found stuff with Scott Minnick's work, and you kind of dismantled that. And like you said, you really loved his paper. Well, by the way, that's another area we agree. We really like Scott Minnick's work.
That paper, for people who don't know, Scott Minnick, who I don't know if he's still there, but at the time he was at, I want to say University of Idaho, I think.
He's still there.
And he's still there. And so he did a bunch of work on the Lenski sit plus stuff and basically dug into it and found like the sequence of mutations that had to lead to that trait. And I love that work because he showed that it's irreducibly complex.
Yeah. See, okay.
Great work.
See, again, we agree. So, all right. So now.
Okay. So we can nerd out about that all day. So let me bring this back. So I corrected the first part. So it's some instances of de novo origin now, and then the second part of that is because it's two kind of qualifications. It's some instances are really unlikely, and the number, if you take almost 4 billion years of evolution, the number of such events that would have to happen, you combine the improbability with the number, and that makes universal common descent not feasible. How's that? Is that part fair?
No, that doesn't represent what I'm putting forward. Okay, so let me explain why. I've told young Earth creationists, because I tried to defend young Earth creationism, I said, look, guys, we don't have, and some people have been really angry at me, and I said, look, because there are old Earth creationists, I think they have a valid point. I don't think me trying to teach the next generation, I said, being scientifically responsible, I said, I don't, if someone with my physics background thinks it's challenging, how much more anyone else? I said, you're going to have to at least settle for now, working with the hand that you're dealt. I don't think the case for young Earth creationism from a empirical standpoint is as strong as I would feel comfortable. I can accept it by faith. We accept some things intuitively. And so I'm saying, look, I'm working from a model where I'm not going to criticize universal common ancestry. Until you can prove the Earth is young, you're not going to do it genetically. So I'm trying to give my reasoning here is like, all right, even in mathematics, you can come up with a true statement, but if your method of proof is illegitimate, the professor's going to say, well, look, we already agree that's true, but you didn't demonstrate it. You didn't go through the right process. I cannot, in good conscience, say that I could take the protein orchards and say that undoes universal common ancestry. That's so
Okay.
It would be it would support Behe's notion of intelligent design, or even Stephen Myers, where you have Stephen Myers suggested miracles along the way.
Okay. So I want to make sure I'm clear about this then, because so you, like I said, you've been talking about this on various other places. You've done some interviews on this. Recently, you were on the Real Science radio show that was on May 9th that aired. And you said in that show, you said, these are direct quotes I wrote down when I watched that interview. No common ancestor for, sorry, this is not a direct quote, but the kind of idea you put forth was that no common ancestor for proteins defeats evolution. And the direct quote was when you mentioned the Aaron Ra quote, when he said there's no effing, common ancestor for proteins. You said he doesn't realize that he just said, quote, evolutionary theory has just been defeated. So what do you mean by that, if not universal common descent?
It is the mechanism. So if we look at Michael Behe, it is the idea that there is a designer. So I was criticizing not common descent, but the idea that where evolution is represented as showing that natural forces can create the complexity. Again, Darwin's Origin of Species, Chapter 6, Organs of Extreme Perfection and Complication. And that's actually been the center of a lot of Michael Behe's work. The notion of irreducible complexity came about that. So unfortunately, sometimes when we talk about evolution, the way that we're using the idea, people may be working from different definitions. And specifically, somewhere else in that interview, they said, does this prove, Doug McBurney said, well, does this disprove common descent? I said, no. I said, no, it does not. I said, it just shows that even if you assume common descent organismally, you'd still need miracles along the way. My point is, you need miracles along the way. Now, the stuff where you said you need many de novo origins and all, I never even considered that. I look at it more from the difficulty of co-opting parts. If you can't even co-opt existing parts, how much harder will it be? And then also that selection tends to destroy versatility. You can't build a microbe into something as complex as a human if the dominant mode of evolution is genome reduction. And even though I don't agree with Kunin's proof, he had the right title. He came to the right conclusion for the wrong reasons. And I quoted that. So I'm very concerned. I could not in good conscience say that natural selection could evolve this. And it was Minnick's experiment that really started to drive this home. And let me see if I have a picture of that. If you could bring up the screen, since we talked about Minnick.
There you go.
So there are like 80,000 generations in Lenski's experiment. And this particular protein/gene/protein, the DCUS, it had a 5-pair base pair deletion. It could not rebuild it even after 80,000 generations. So Dawkins Weasel says natural selection, it's going to solve all these problems. It will mutate it, and then, it'll preserve on the way to rebuilding things. I said, this is not going to work. I don't expect it to ever work. It's a frame shifting mutation, since it's not divisible by 3, 5 base pair deletions, not divisible by 3. And it's just going to get worse and worse. This does not, you can't take those equations of population genetics that we were showing earlier and demonstrate that this is going to reconstruct a gene, even though it's only 5 base pairs away from getting the right solution. You could take this down, and this is more of an example of why I don't think natural selection, if it can't even recover a gene that's almost there, it's going to have a very hard time constructing something as complex as some of the genes that I have studied, like the zinc finger and the collagen. Now, there might be some transcripts out there that come from the non-coding regions that could be spurious. They could do something. And I'm not even going to argue that, I'm not even going to say that they aren't de novo genes. They might well be. I wouldn't put it in the same category of complexity. And so your other argument about the number of de novo origins, I haven't even considered that. I was just looking at this experiment and then some like it, particularly Lenski's experiments, where there was a lot of gene loss. In all the cell lines, it was losing genes. And so the way I liken this is to a hiker. He wants to move faster, so he dumps all his gear. So in that environment, he can, when he becomes a specialist, he becomes better at that specialization, but he loses generality. And if you have different selection pressures over time, you could actually induce a lot of gene loss. That's a testable prediction. And I just don't see natural selection being able to overcome that. So I'm going to even grant that we have some de novo genes that keep emerging all the time. It's the level of complexity that bothered me. I can't speak for all gene families except the ones that I've studied.
Okay. So now I feel like I understand the argument better. It's not an argument against universal common ancestry. It's an argument against naturalistic, undirected evolutionary processes.
Right. And, because naturalistic is a metaphysically loaded word.
We'll say
far from normal expectation.
I would say undirected processes, right? Processes that don't have a conscious intention behind them. Right. So a couple of things I want to toss out there and see where it goes. So first, you're right that natural selection is a mechanism that essentially streamlines genomes, right? Natural selection results in the loss of things over time. It's not a creative mechanism of evolution. It will make you better in your current environment, but if your current environment is a certain amount of glucose in a flask that runs out by, the afternoon of that day, yeah, you're going to get rid of some, extraneous stuff to make you better at competing in that particular environment. In other environments, what works best is going to be different, and natural selection is going to lead to different things. There are other evolutionary mechanisms that go in the other direction, right? And it's the interplay between all these mechanisms. So, for example, we know, there's that famous paper, I think you showed it, gene loss is a dominant mode of evolution. That's true for a lot of different organisms. But what happens along the way is a lot of duplication events where you have whole genome duplications or chromosomal duplications. So you end up increasing the amount of stuff you have to work with, and then selection streamlines it. And then you increase it, and then selection streamlines it. And as an example of this, we can just look at vertebrates, for example. When you go from the invertebrate chordates to the vertebrate chordates, and then to the gnathostomes, the jawed fish, you see successive rounds of whole genome duplication. And that leads to increasing complexity in their developmental patterns. And these are incredibly common mechanisms that increase just the raw amount of stuff in the genome, right? And then what happens, you have these big increases, and now the genomes are inefficient. So natural selection cuts them down. But you have this pattern of duplications, increases, and then cut it down, and then increase, and then cut it down. And the overall pattern is that gives the organisms more flexibility to adapt to this or that different environment, right? And these are the kinds of things that have happened as different clades have specialized over, their evolutionary history. But that's very theoretical. I think it's better to focus on like concrete examples of this stuff. So like you've said that like de novo gene origins are a thing. And yeah, I agree. Like we have lots of examples of de novo genes, right? There's some really famous examples, like the one that everyone's probably heard of is, if you've heard of one de novo gene, the one you've heard of is the antifreeze protein in the Arctic fish, right? Right, Sal? That's one that everybody knows. Behe talks about that in one of his books. I think that's prominent in Edge of Evolution. Basically, like this is a new gene. It doesn't have a common ancestor in any other group of fish. Like this is a thing that evolved completely new. We have examples of that in humans. We have examples of that in rodents. So you've got like rats and mice each have their own de novo genes. You've got lots of them across different ape groups, yeast, plants like rice. There's a bunch of studies of de novo genes in the rice family. So we agree, I think, at least in principle, that like de novo genes are a thing that evolve. That's a thing that can happen. So the question is what you talked about, the level of complexity. So here's where I get on my high horse. What's the threshold and how can we measure it? How can we quantify the complexity and then say, here's the threshold that can evolve. If you exceed that amount of complexity, you need some kind of intelligent input in order for that thing to have evolved. Where is that line and how do we know it?
No, those are, by the way, if I may give you a bravo for what you just said, I actually didn't even study much of the antifreeze gene. There's so much literature on both sides. I've not been able to survey it. I will point out that when you do a gene duplication or even whole genome, you're still talking about homologous genes.
Mm-hmm.
I'm wanting non-homologous.
I got you. Yes.
Okay,
yep.
So as far as the threshold, so I'm not. This is intuitive. So you ask a pointed question: how much is enough? For me, like 10 to the 500th power, something on that order. And I have some examples also where I have one specific example that converted someone from communist China. She was probably an atheist or agnostic, became a creationist Christian. And it had to do not just with de novo complexity, but also co-opting a pre-existing part. So if it's difficult to co-opt the pre-existing part, and I'll diagram this up, by way of extension, it's going to be hard to make it. And I'll just, if I could show this, I'm not quite there yet. Let me get to the right slide if I have it. Oh my goodness, I hope I have it here. Yes. Okay, so if you could bring this up. This is the spelling of the topoisomerase protein, and all the colorized areas are critical to its functioning. One thing I do know, there is a section here, it says DQ, like Dairy Queen, Dairy Queen. Joe Dewey's dissertation said you knock out that's the magnesium binding site. Just those, even one letter in that region, topoisomerase is gone. And there are probably critical domains here, like the ATPase domain. One of interest is here, the bipartite nuclear localization signal. I don't know if people can see where I'm highlighting. It's that red area here at the very, kind of toward the bottom. And what would happen is if you're transitioning from a prokaryote to a eukaryote, all of the homologous proteins in the prokaryotic form have to have inserted simultaneously, especially the ones that are life critical. See, all of them, but particularly the life critical ones, they need a nuclear localization signal. That's what it's spelled like. That probability is kind of remote, even for, even smallish passwords are not easy. So if we're talking about an isolated protein, maybe, it's not quite astronomical, but when you're talking about all the life critical proteins going from prokaryote to eukaryote, we could do the odds, and even with generous numbers, that would be like really far from, astronomical. Change tan, she's on the left here. She was picked by Nobel Prize winner George Smith to teach molecular biology. He was very impressed with her. She's one who made that transition. So I'd say from a qualitative standpoint, that was enough for one person. we could throw out numbers, but it's like, well, then those numbers are based on, how good are your assumptions, and at that point, it's still you could take that down. It's still kind of intuitive, but so I'd say at a personal level, how improbable is it for each person to say this probably isn't, this is astronomically far from normal expectation. So when I started to examine that, I was just like, well, this is remote. So the other thing that I had studied was even co-opting zinc fingers when you're shifting domains, even though this is homologous. Because, and I cited a paper earlier, it's very interesting that, let me see if I could bring it here. It was Structure and Function in Naturally Evolved Denovo Proteins. I only found this one today, Dr. Dan, and I couldn't get it through my university access. And because we are trying to make, the one area that I have in passing studied is designer transcription factors for pharmaceutical applications, where you can kind of you're trying to modulate the genome. And so they're trying to make these transcription factors. And one thing that struck me about this paper, it says the existence of de novo proteins seems at odds with decade-long attempts to construct proteins with novel structures and functions from scratch. So anything like in that industry where they're using things like zinc fingers or any of these other complex proteins where you have multiple interactions and connection or binding partners, they know the difficulty of making a pharmaceutical to kind of modify how an organism functions. And they're like, well, okay, we now have kind of the requirements and thresholds. I know kind of, I have a slide here of the complexity of zinc fingers. And I did kind of I did an examination of this, and I said, we also have the waiting time problem with this. These are astronomical numbers. Even if you reject John Sanford's waiting time problem, you still have Durrett and Schmidt. And, Durrett and Schmidt were assuming how much selection pressure there could be. I said, that assumption's not based on the actual way a zinc finger works. If you're talking about a zinc finger transcription factor, I said, this is going to be, okay, so can you take this down? I wanted to answer I'm trying to answer your question directly. John Sanford, just before one of the projects he assigned, but we never got to it, he said, why don't you take this zinc finger problem, estimate the odds, put it in a waiting time problem? And I said, well, I'm moving on to other things. And so the question you posed, it's still an outstanding problem. I'm just saying from an intuitive level, I don't have an answer. It may not satisfy you. It has satisfied others kind of at a personal level. And so I think the question you asked is very pointed. It's worth trying to address. And I don't have a number to give you. What is the threshold? I can just give it from an intuitive level. And then that particular paper kind of also articulated kind of what I was seeing. This is, when we're actually trying to make substantially complex systems, it's like, why does it seem at odds?
So if I understand you then, because my question was, how do we quantify this threshold? Where's the line? How do we know? And the answer is, we can't yet quantify it. We can't assign a specific threshold, but you can intuit that certain things are too complicated to have evolve through undirected processes because they have a bunch of different components that need to be quite precise, and they all need to operate together. You said critical things need to be there simultaneously was the phrase I wrote down as you were talking. And that is sufficiently improbable that for you and for some other people, you say, okay, that's too much for me. I don't think this could evolve through undirected processes. Is that a fair representation of kind of summary of what you just said?
Yes. And
Okay.
I just want to say, okay, if people believe in a multiverse, it's like it still could be evolved by undirected process. It just still be far from the normal expectation. I'm trying to avoid metaphysical language because I don't like going. But I mean,
Well,
I have tried to go through the numbers myself, kind of back of the envelope. I wouldn't present it in a peer-reviewed paper at this stage, but the numbers are okay. So where I go is like, okay, let's at least.
well, here, hang on. Before we move on from that last thing, can I just kind of respond
Sure.
to? I want to make sure that I was accurate. I was kind of on the same page as you. Now, let me respond to what you just said. So, with the big figure you showed with the sequence that had the different regions that highlighted in the different colors, showing that they're all required. I'm not familiar with that specific experiment. But a lot of times the way those experiments work, and correct me if you happen to know and I'm wrong, correct me. But oftentimes the way those experiments work, where we say this region is critical, is that we delete that region and we see if the protein is able to do its specific function. I'm not, like I said, I'm not familiar with that case, but I'm familiar with several other examples where we can say this specific function requires this specific region. And the way we do that is we make a version of that gene that's missing different regions. And oftentimes you do like a slide, like a kind of a sliding window kind of thing. And you kind of hone in on the specific required regions. Now, that's one way to identify the regions that are required. But it is, so first of all, do you know if that's what they did in that experiment? That's typically how that's done, but I don't know that specific one.
Yes, and I do want to add one thing. I mean, this is relevant. The statistics we develop for one protein are generally not extrapolatable to another protein because the architectures are so different. It's like saying, what's the probability of making a spark plug or a piston? It's just like, it's so hard to just like, well, how do I just want to, so that, let me show that diagram here. So these were,
There you go.
if it's not knockout, there are also various ways to chemically inhibit stuff. But also at an intuitive level, if you don't have a part that's contacting the DNA, because the topoisomerase cuts DNA, you need to have a region that contacts the DNA. You need to have a region where it binds ATP. So if you could say, well, maybe it doesn't have to be exactly in that location, but you still need an ATP binding site. You need a nuclear local. So there's some minimal parts there that you could just start to take inventory.
Gotcha.
And there is one picture that I've omitted. There are a huge number of, you could take this one down. There are like 38 disulfide bonds to help it structural. There are 60 post-translational modifications. And I'm just like, when you start to add these up, it's like, okay, this is why some of them just became spontaneously creationist. It's like, well, they didn't necessarily go through the formality. So I could say that, how many disulfide bonds are you going to knock out before this thing falls apart?
Right. Okay. So first of all, let me just, before we move on, let me just pull up the super chat briefly from My Unfinished Journey, $5 super chat. Sal, I appreciate your willingness to step onto scientific turf to make a young earth creationist case, as well as your civility in doing so. And I second that. I appreciate you being here, and I always enjoy our conversations. Thank you, My Unfinished Journey, for your chat. That aside, so I asked about the way that experiment's done because there are kind of two different ways of approaching this question. You can look at it and say, okay, we have the parts, and then we have the sequences. And a lot of times you need specific parts, you need specific components, you need this, domain, you need this DNA binding loop, whatever, right? You have these specific parts. But when you look, and you can document that you need those parts by doing knockout experiments, you can remove the specific parts and show that you need this or that for the function to happen. But the other way of looking at it is doing substitution experiments, where you change the sequence and see if it still does the function with a different sequence. And when you do that, you often get a very different story, because what ends up being the case is for a lot of these functional regions where you need a specific domain, you need a specific binding site, the specific amino acid sequence is not itself tightly constrained. Maybe there's one or two amino acids that need to be a specific thing, but within that region, most of those amino acids have some kind of flexibility to them, where maybe it needs to be basic or it needs to be non-polar, but as long as it meets some kind of set of criteria, any set of a number of amino acids are going to work approximately equally well. And this isn't just me, like, talking off the top of my head. We have robust experimental evidence that this is the case. So, like, you can, for example, it's not a protein, it's a DNA sequence, but it's the same kind of logic, is that you can randomize the nucleotide sequences for a bacterial promoter, and you can just make random sequences that are approximately 100 nucleotides, about the length of a promoter, and huge percentages of those sequences will work as a promoter. Some of them will work as well as the wild type promoter. And it's just randomized sequences. You're just generating millions of sequences and just trying them all and evaluating function. And you can do this with proteins too. And we could also look at examples in nature. So one of the examples that I know really well for this is HIV. And Sal, I think you've probably heard me talk about this before. HIV VPU, and it has this specific activity called tetherin antagonism, and it requires a specific domain to do this. And this trait has actually evolved twice in HIV. It's done a completely different way in the various SIV viruses and the various SIV proteins. Simian immunodeficiency virus, it does the same thing in their respective host, but it does it a different way. And in humans, so HIV1 group M does it a certain way and has a couple of domains that are involved. And there are 7 very specific amino acids that you need. There's like, you need boom, boom, boom. And then on the other side, it's like 4. It's like 3 and 4. And you need those specific 7 amino acids. But then there's another type of HIV. I think it's HIV1 group N, but I could be wrong. Everybody chat, don't quote me on that. I think it's group N, but I might be wrong. But it's one of the other variants of either HIV1. It might even be HIV2. But it does the same function. Instead of using those 7 specific amino acids, which are tightly controlled in HIV1, in this other HIV1 group M, in this other one, it just uses a string of negatively charged amino acids, and it accomplishes exactly the same thing with a completely different sequence of amino acids. And my point here with this is that we can look at a specific sequence like Douglas Axe did in his famous 2004 paper, right? We can look at a specific sequence and kind of try to work forward to that sequence. Say, what's the probability of this specific sequence happening? But I don't think that's a good way to calculate the probability of a certain thing evolving or a certain thing being able to evolve, because we know that that specific sequence is not the only way to accomplish that function. So a better approach is to go in the opposite direction and just look at randomized sequences and see how many can accomplish that specific function. And when we do it that way, we find it's really easy to get all of these specific functions. So that's my first kind of counterpoint to this, that it's, in addition to not having a specific threshold, just intuitively, we know from direct experimental results that that threshold, wherever it is way higher than I think you're presenting, because we know there's way more than the ways that exist to do each biochemical function.
So let me at least support half your point.
Oh, go ahead.
Again, bravo. I think you made your case very well. So the reason I say that is in bacteria, the topoisomerase is implemented differently. It's actually a heterotetramer, the gyrase plus one other thing. So there's not one way to implement this DNA unknotting. That actually supports what you just said. And this is why I was very fascinated by the zinc finger, because this is like, if I can make an analogy here, as analogies go, there are infinite number of ways to make a key and lock set that match. Infinite. It doesn't make it highly probable, though, that you're going to be able to make a key and lock set randomly. And in the case of the zinc finger, which specifically looks for a sequenced partner, either a DNA, an RNA, and in some cases another protein, we usually know zinc finger is going after DNAs. So one thing I saw in evolutionary literature, they're saying, look at the coevolution of the zinc finger in the promoter regions, or the, like ERVs. And I just like, well, whoa, this is again that lock and key problem. You have to Yeah, they said we discovered this new mechanism of, this new phenomenon of coevolution. Right away, that was a concern for me, because if you evolve one, you have to simultaneously evolve another. And I would agree there are an infinite number of ways you can make zinc fingers, and infinite number of ways you can make promoter regions. I mean, hypothetically, it doesn't make it likely that you're going to be able to make a pair that is physically located close enough to the coding regions that it's going to work. And I've in the last 2 weeks, when you sent this invitation to me, I was actually thinking about that problem. And we're seeing that problem in the pharmaceutical industry as we're trying to make designer transcription factors. It's not easy. If you're given a promoter target, it's pretty difficult to make a zinc finger that's going to target that specific sequence. So again, I think your criticism of my presentation on topoisomerase was, I'm going to give you really good credit on that. Okay, so again, this is the value of interacting, because now you've improved my edit. You've given me free editorial review, and I'll try not to make that mistake again. But in terms of the nuclear localization, can you bring that thing up again? This nuclear localization signal does not have to be exactly this way. It has to, it's not exactly that sequence for every protein that has to have, that is nuclear localized. But it's a small space, because otherwise it's not going to work. You're going to let everything through. You want to be very discriminating. And you could take this one down. So this is a testable hypothesis. If we see that different organisms implement the nuclear localization signal differently, this again is like the lock and key problem. There might be infinite ways of doing this, but it doesn't really improve the probability of being able to manage all these. If you had to co-evolve all the nuclear localizations across all the proteins in a particular organism, like say yeast versus humans, I would say that would, at least, we could put numbers to that and say, look, this looks pretty improbable. And that is something I just haven't pursued. I'm actually going into.
You should pursue that. That's
okay.
That's the whole argument, is quantifying that threshold is that's the crux of the whole argument here. Because kind of like while you were talking, I thought of another example of this where different sequences can do the same thing. And we've done experiments where you can take, for example, genes out of fruit flies, and you can put them in chicken embryos, and the sequences are obviously quite different from each other, but in terms of the regulatory network, they fill the same spot in that network, and it works, right? You could show that they work. That would not be the case if these sequences were hard to evolve, right? If there was a specific sequence that needed to be there and only that sequence worked, it wouldn't be possible to do those kinds of things, because the same protein across different organisms has, in some cases, quite different sequences. But we've shown that you can mix and match and use different combinations in different organisms, and it still works in many of those cases. But another aspect to this, there's one big thing I want to highlight here, but I just want to say kind of a small thing. In terms of evolving these interactions, protein interactions and protein nucleic acid interactions are very fuzzy, right? Because these molecules have regions that are hydrophobic and hydrophilic. Pretty much, like, that's just going to be the case because of the types of molecules they are, right? DNA has that. Proteins have that just based on the structure of amino acids. You're going to get some level of association with relatively little sequence specificity required. And the question is, what threshold of association do you need for it to kind of assume a functional role in terms of regulating gene expression, for example, right? And that we can quantify that. And actually, in the chat, I'll just highlight something somebody said in the chat that scooted by here. Here we go. This was from Answers in Atheism. Zinc fingers, sometimes you only need 3 out of the 20 amino acids in that particular region to be the correct ones in order to associate with the DNA strand. So you have a lot of flexibility in terms of the degree to which these interactions can occur very robustly or very transiently within the cell. I want to give you a minute to respond to that. And then there's one other thing you said that I really want to highlight that I want to get to. But first, let me give you a minute to respond to that if you want.
I totally dismiss that last comment. And it depends on whether we're talking about monovalent versus multivalent. So it can bind if we're talking about something like CTCF, binding sites that are multivalent. You don't necessarily need a lot because you're trying to control a lot of the binding toward signs that control the chromatin looping. But something like transcription factor 3 alpha, it's going to need a very specific. I mean, it's going to search out. It binds not only to the DNA and RNA. So you can't generalize one trivial example and say that counts for all of them. So I totally dismiss that. Thank you.
Well, okay, but you were using zinc fingers as your example. That's your example. That's not mine.
Specific zinc fingers for a specific transcription factor binding site. I was not talking about CTCF binding sites, which are multivalent and go to all sorts of places that aren't a specific sequence. So, I mean, that gives me an opportunity to clarify here. But when I talk specifically about designer transcription factors, this is the problem that they're having. It's not that easy. If it were that easy, they would be making these, these designer transcription factors rather easily. And it's still a challenge. I mean, if humans are having a hard time with all their computational and biophysics, I just don't see that, random mutation is going to be able to give it to you.
So how do we quantify which ones are, like, it goes back to this question of quantification, right? How do we quantify where that line is, which you said we haven't done the work. And I think I'm often critical of like intelligent design creationists for like basically pretending there's a big robust research program when there really isn't. I would like, this is an example of like research that somebody could actually do and like do the math on this and say, here's a threshold, we do 1 million of these, we randomize the sequences. For this specific one, X fraction of 1 million associated with the DNA. For this other one, it was Y% of that million, right? Like that's an experiment somebody can do.
Well, this is where I have to sympathize with your criticism here. There was an internal discussion. I think I could share this. It's like, okay, we have limited resources. Are we actually going to be spending money on experiments? We're going to let other people spend money. And so some of us said, let other people spend the money. So a good example is ENCODE. We were so thrilled with the ENCODE project. They did a lot of what we were hoping someone would at least try to do. So as far as what we would do for research is just monitor what the pharmaceutical industry is doing, because they are kind of helping us give the numbers. Then also that experiment where Scott Minnick analyzed the Lenski experiment, 80,000 in the DCUS gene was not recovered. I said that was pretty, like, I didn't know about that. I'm just like, wow, even after 80,000 generations, it didn't recover a gene, and it was only 5 base pairs away from being functional, and it couldn't recover it. So I would say, how do we draw the line? I think we just wait on the experimental data. We look at the pharmaceutical industry. A lot of ID proponents, by the way, are in the pharmaceutical industry. They won't make it in academia, obviously, if their sympathies are known, but they found a home in the pharmaceutical industry. So I'd say, how do we gather the evidence? I don't think it's going to be so much by theory, but through experimental practice, we start to detect that. So for me, that one where Scott Minnick was showing that genes were being deleted, and particularly genes are not being able to be recovered through natural selection, at least, we could put numbers to that. Okay, 80,000 generations, this is the mutation rate. We have a lot of data on that. So these experiments are expensive to carry out. And so when you talk about the robust ID research program, I'm not going to defend that there is or isn't. I'd rather not engage in that debate, and I'm going to get myself in big trouble if I give my real opinion. By the way, I've been criticized for, okay, I'm just going to come out of the closet here, and I already have. I said, I don't defend ID as being science. I think ID is outside of science. If you want to say it's science, I'm not going to argue with you, but I'm not going to make that case. And I'm not going to say we have a robust research program. Casey Luskin can get on me. He said, well, that's fine, Sal, you could say that. We're still friends. So, so some of this you might say is metaphysical. And I'm just like, hey, I'm cool with that. I've tried to argue the scientific side. I'm not going to make the case formally that you should believe in a designer from any of this. I'm just saying this is kind of my personal decision, and I share that with other people. But in terms of the strictures of science, I'd say I think the criticisms you fielded are very good. On the other hand, I'm sorry to be a little pointed here. The sword cuts both ways. I think natural selection as a paradigm is not delivered. The way Richard Dawkins has advertised it in Blind Watchmaker, I think that's been falsified on many levels. And the fact that the third way of evolution has kind of emerged, and Jerry Coyne's kind of livid with that, it's kind of showing that, the Darwinian viewpoint of natural selection evolving organs of extreme perfection and complication, I don't think that's been fulfilled.
Yeah, I mean, I agree, because Darwinism is from 1859, and I agree that doesn't explain stuff. We've come a long way since then, but that is neither here nor there. So the big thing that I want to get to here is I want to just point out, and I want to ask you to explain where you're coming from here, because over the last little bit here we've been talking, you said two phrases that I wrote down. You said critical things need to be there simultaneously, and then later on you said if you evolve one, you have to simultaneously evolve the other. Speaking of two proteins that interact with each other. I know you have said that you don't like to use irreducible complexity as an argument. But how is what you're articulating not just an irreducible complexity argument without saying the words irreducible complexity?
Oh, no, I'm really glad you're giving me the opportunity to clarify. I would say you have to add qualifications to make it a workable argument, because exactly what happened when you just throw out, oh, this is irreducible complex, so it can't evolve, and you don't put any statistics to it, it'll be like a replay of you and Altangelo, where I gave you the win on that one. Okay. It has to be specific to the system. So I'll say, yes, it's irreducibly complex, but with these additional qualifications, we actually go into the mechanical biophysical details. And, I think some of your criticisms of my viewpoint are very fair, but I just have to articulate this. The sword cuts both ways. If evolutionary biology is going to, if at least in the popular press, claim that this explains everything, it's like, no, definitely not, because then that side has to answer those statistics. Now, with people like you and others who are very thoughtful thinkers, they'll say, look, these are open questions. I respect that a lot. Okay. So I'm just saying that the sword cuts both ways. And I'm sorry, what was your point trying to get out of me?
I was saying, how is this? So the question was, how is this argument, and specifically like critical things need to be there simultaneously? Right. How is that not just Behe's irreducible complexity argument? That's what that is, right? You have a system composed of multiple parts that are all required for that system to work.
It is okay. It's the degree of improbability. The degree of improbability. You could see like that nuclear localization signal is not a trivial sequence in terms of its length. We don't know exactly the sequence space, but it can't be a large sequence space for just one protein. But if you have multiple proteins, you multiply the improbabilities. Say if it's even 10 or 100, then it starts to become astronomical. And I think you rightly pointed out that might be a good research area. And particularly if we find that the nuclear localization signal is different among species, it's like they would have to co-evolve. So when I specifically mentioned a paper, coevolution, there was a paper that talked about the coevolution of zinc fingers and ERVs. I don't know how I stumbled on that. I was just kind of rather shocked. We were saying, oh boy, the ERVs had to evolve with the zinc fingers that bind to them. And I'm like, that was pretty interesting. And these are things. This how does it distinguish from what Behe says? I'm saying Behe is the way it's being used by ID proponents is too broad. You have to say some of these irreducibly complex might be able to give you the improbability you want. I definitely would not say all. And Otangelo is defending all, and I'm like, you're going to get slaughtered. And I credit Dr. Dan with the win on that. I said you called him out on the problem with that viewpoint. So I would say some, and let it be the proteins and systems that you're familiar with. Because I could talk to these researchers and they'll say, yeah, this is why I'm kind of an ID proponent. I researched the insulin receptor, or I researched topoisomerase, I researched zinc fingers, or any number of other things. If it's in your field of specialty, then talk on that.
So, okay, so I was taking notes as you were talking there, and what you said is the difference between kind of canonical Behe irreducible complexity and what you're talking about here is that you have to add qualifications, you have to put statistics on it, is what you said.
On specific systems.
Right, it has to be within specific systems, and you have to assign statistical probabilities within the context of those specific systems. But here's the thing, and I keep coming back to this, and this is very similar to what I was talking to Dr. Behe about. This is the same thing. Has anyone calculated those probabilities? What are they? How do we know? Where is that threshold? Right? It's the phrase you said is the degree of probability, but where is that line? If we can say this protein or this system can evolve, this one can't, we have to be able to quantify that. And the way you described it was some irreducibly complex things might give you the improbability. Without any numbers, that's an unfalsifiable statement. Now we're just playing whack-a-mole, where if I show you empirically that a protein that's 100 amino acids interacting with another 100 amino acid protein, if I show you experimentally that can evolve, okay, that sets the floor, right? But then anything above that we haven't documented. Well, what if now we show higher levels of complexity? There's always going to be more examples that you can point to and say, this is more, that's more. But without like an empirical basis for making that judgment, it's subjective.
Well, we do have empirical basis for nuclear localization signals. I showed you one here. This is about 20 amino acids. So let's say you only need 5, but then there's an improbability with that. Okay, so let's say it's something like 20 to the 5th power. That's a lot to resolve. But then if you have to spread that out to like 100 proteins, this is complex. And not to mention you need the important complexes and all the nuclear pore complexes. You start adding all those up, it doesn't take long before you're already at astronomical, even if you give the most generous numbers. So I will take your criticism and say, if you want me to work it out, I will. It's going to be a laundry list. And I think people are welcome to try to criticize it. I think you made a very fair point, and this is why I really like you. I think you try very hard to represent the opposing viewpoint fairly, and you give good challenges. And as I've said, you've corrected me many times, and some of your challenges have resulted in me amending the way I think. So I'm not going to dispute that. Why don't I write something up? It probably people will just, I don't know how they're going to react, but at least I'll give it a stab. And I think that's you've given something that I think we could look at for nuclear localization signals. I can also think of translocation, which is the stitching of transmembrane proteins into the cell membrane, zinc finger proteins, and then you add waiting time. But I got to say, at least for the fans of evolution at the popular level, not your level, Dr. Dan, I'm not including you in that group, who are arguing that evolution's proven. I'm just like, the sword cuts both ways. You have to come up with your statistics to show that natural selection can build these things, random mutation, natural selection. And I definitely think I've made the case that has not been done. And we can also then criticize whether those assertions in the popular press are scientific. I would absolutely say they're not. except as far as speculations are allowed as part of science. But I wouldn't say it's at the level of confirmation of other scientific theories like electromagnetism. So I think your criticisms are fair, but in defense of my viewpoint, I'm just like, well, this is the reason we don't believe in evolutionary biology, because we don't feel that they have given us convincing proof that it evolves naturally.
I think that work continues to be done. I mean, there's every year there's more work on both de novo genes that we're finding, and and I will say de novo genes, within groups that creationists agree are share common ancestry. And if we want to hypothesize tweaking along the way, then now that's gets into the realm of unfalsifiability. Right? There's no way to evaluate divine intervention that looks like an evolutionary process. I also want to say that in addition to the work on de novo genes, we're also doing this work, kind of that shotgun approach I described earlier, of just like trying out a whole bunch of sequences, and we keep finding things that work where we have a wider range of sequences that do different functions. I think we're coming up on a good place to end, so I'm going to say a couple more things, and then I'm going to turn it back over to you, and I'll give you kind of the last word here, and then we'll call it a night, and that'll get us to about 90 minutes of chatting, which I hope everybody appreciated all of that. So a couple things I just want to say here is that specifically with the nuclear localization signal, I don't think you're kind of giving credit for how variable that can be and just how widespread it is. It's actually found outside of eukaryotes. You actually have some archaea that have a nuclear localization signal, even though they don't have a true nucleus. They have some archaea have kind of a similar structure, but it's not really a true nucleus. But it is, I think, a more variable thing and more widely found thing than you're implying. But that's a kind of a small point. A bigger thing here, I think, and a kind of a bigger problem with the structure of this argument is that you're taking an existing localization signal and saying how hard must it be to evolve this thing, but I don't think anybody thinks that the current form of like something like that is the original form of it. It would have first appeared in a more rudimentary form. And that's where kind of those fuzzy interactions can kind of come into play, where it doesn't have to be as precise as it is now in order to do some little bit of that function. And that's the hard step, just getting a little bit of it. Once you get that little bit, then natural selection is really good at optimizing, right? That's where natural selection is actually very effective. And I think we would agree, maybe we would agree on once you have a function, natural selection can tweak it in the face of mutations. So the last thing I want to say, and then I'll turn it back over to you and I'll give you the last word on all of this, is that I think the key thing here is quantification. What you're proposing is essentially irreducible complexity with a threshold, rather than saying if it meets these criteria that Behe kind of articulated. It's not just those criteria, it's those criteria in the context of a specific system with a specific probability threshold. And beyond some threshold, we have to evaluate that system as being unable to evolve through undirected means. I hope that's a fair representation. And the linchpin of that whole argument is quantifying where those thresholds are. How many components does the system need? How stringent are the requirements for the components of that system? What is the probability associated with those components? And at what probability does it become too improbable for evolutionary processes to have done it? And I like quantifying that threshold is the thing. And I think we agree that to date that work has not been done. And that's, I think, the weakness in this argument is you need to be able to quantify where these, I'll use your phrase from the beginning, the statistical miracles, where's the cutoff for that? That's what we need to quantify. And I think that's really the big thing. And if you can do that, you'll have a much stronger case. But that work has yet to be done. And I don't think creationists should wait for the pharmaceutical industry to do that math. I think if you all want to be taken seriously, get in a lab, do the work, get us those numbers, and then put it out for peer review, and not in like biocomplexity or something. Put it in, you know, a real journal, and like, let's see how it holds up. I think that would be great.
I'd like to thank you for giving
Over to
me.
you.
Well, it was very kind of you to give me the final word. We both agreed that selection can result in gene loss. So it seems to me speculation that it can build. So the sword cuts both ways. If you're saying I didn't prove my point, I think even more so, I've shown that evolutionary biology has absolutely not proven their point, that this can emerge through evolutionary processes, some vague definition. And when you're describing the nuclear localization signals, you're using speculation to plug the gaps. That's fair given where we are. But if you're going to hold me to these standards, you need to, I think it's only fair that we hold the evolutionary biology community, if they're going to make assertions and publish peer-reviewed papers, they better deliver. And what's happening, the reason there's been kind of a revolt that I'm seeing, kind of like people like Change Tan, it's like, well, okay, maybe I haven't totally, she or I or ID community may not have totally proven our case, but we think that evolutionary biology has done far worse in proving their case, because we've identified all these areas, all these problems, and it's just like not believable anymore, because we come to evolutionary biologists for answers to how do you build this complexity, and it's filled with speculations and made up numbers, even more so than our side. I cited specific experiments of gene loss, and then genes not evolving, even when it's only 5 base pairs from getting to where it needs to be. Scott Minnick's example. Nuclear localizations have to be sufficiently specific to be able to do discriminating things. Same for translocation processes. I talked about the lock and key problem. There are infinite ways to make these pairs, so it doesn't make them highly probable that they'll exist in any physical location closely. We're talking about the ID proponents being taken seriously by the scientific community. What I'm finding is the scientific community is not taking evolutionary biology very seriously. It might be only, they might be there metaphysically, but certainly the science is not on par with other scientific disciplines. And I'm seeing that in the research teams, like in the pharmaceutical industry, they like biochemists, they like engineers, computational scientists. It's the evolutionary biologists that are kind of not there. And I'm sorry to be harsh because that's your field, and I know it's something you value very much. But so the sword cuts both ways, and so when we see just kind of on an intuitive level the organs of extreme perfection and complication, and we see that evolutionary biology is just using speculations, and even their definition of fitness is so faulty it doesn't even work to explain this. It's just hard to take that seriously. So, the sword cuts both ways. And right now, both sides are arguing kind of intuition. And I think what you put forward is fair game and a good assignment. ID proponents can up their game and start either doing experiments or at least monitoring them and providing calculations. And I think the fact that you thought that these would be promising areas for the zinc finger localization signals, et cetera, that kind of says, well, if Dr. Dan gives it an endorsement as something to study, I think that would be a worthwhile pursuit. And I really hate having to be so disagreeable, because I love Dr. Dan. And I really, this is the part of the conversation I'm not eager to be a part of.
But it's
I'd rather talk about race cars.
It's fun when we disagree, though.
So I'd like to thank you for giving me a chance to air my viewpoint.
All right. Well, thank you for coming on, Sal. One last thing from you is just if you have any other, I know you've been making the rounds on various, shows and interviews and things. So if you have anything else in the pipeline that people should look out for, anything that we should.
No, this is the biggest Denver KLTT radio. I expect there's going to be something related to the church that has nothing to do with biology. I was in a major lawsuit. Millions of dollars in legal fees from both sides. Already 1 million people have viewed the trailer, so I'm going to be more famous for that than anything I really worked on. It's a legal scandal. We expect maybe 4 million views. I'll be in the documentary. It's called The Real David Platt, so it's going to air in June, and that has nothing to do with anything we talked about here.
I'm going to be on the lookout for that, though. That sounds like a heck of a story. So I'm gonna be looking for that. Okay, so coming up here on Creation Myths next week, we will have a new video. It's going to be a creation contradiction. And then the week after that, so 2 weeks from today, I'm going to rebroadcast my recent debate with Dr. Casey Luskin on junk DNA. And then after that, I have a live show that I don't quite have my guest lined up yet, but I'm going to keep it under wraps for now, but I'm really excited for that one. Yeah, and then I have a couple other things that are kind of further out, but I've got the next 3 weeks kind of planned out. So like I said, everybody, it's a new year. Doing it every Wednesday. I'm almost halfway through the year, hitting every Wednesday, and I'm gonna keep that up as long as I can. So once again, Sal, did you have something you wanted to add there?
Yes, I did have a curious question. Cy Gart is part of your school. Have you ever met him?
I've never met him, so I think it was just he kind of visits every now and then or something. I don't know specifically what his affiliation is.
I was a student at his school down here in Washington D.C., the FAES graduate school. So we do have kind of a remote connection.
Yeah,
I had to ask.
Yeah, and I like, I've chatted with him in the YouTube sphere a couple of times, and I've always had good chats with him. So Sal, thank you again for coming on. Folks, if you don't know, Sal's channel is so there's evidence, and I always get it's evidence and reason or evidence and reasons.
Evidence and reasons is
evidence
my
and
channel.
And I will link that down below. It's not currently there. I will link that down below. And then for longer form type lectures, there's Evidence and Reasons Academy as well.
Evidence Reasons Academy.
Evidence Reasons Academy. There you go. I'll link both of those down below, everybody. They're going to be there within a few minutes of this show ending. So thank you again, Sal, for hanging out with me, chatting about protein orchards and what they mean for evolutionary theory. Hope everybody enjoyed this. Remember to, if you're somehow wandered into this and you have no idea what's going on, please subscribe and come back next week when we'll have some new stuff to talk about. Have a great night, everybody. Thank you all for being here. I will see you all soon. Good night.